Hello. Today we are going to start looking at the nervous system. The nervous system includes your brain, your spinal cord, and all the nerves in your body. We're going to start with just the basics of the nervous system, and then we'll jump into the different chapters on the central nervous system, the peripheral nervous system, and then the autonomic nervous system. So let's dive in. So, what is your nervous system? Well, that's what controls everything you do. It allows you to move from place to place. It lets you know what's going on in the room around you. Like, is it too hot right now? Are you too cold? Your nervous system helps you determine that. And we use electrical and chemical signals in our nervous system to communicate with the outside world, within the nerves communicating with each other inside your body, the nerves communicating with your muscles, with your different organs. They all use electrical and or chemical signals. So all of our nervous systems, they all detect some kind of sensory input. Sensory could be hot, cold, pain, touch. Those are all different kinds of sensory inputs. Or could be a feeling of hunger, tired. Those are different sensory inputs as well. Integration. Your brain interprets a lot. A lot of information comes in, it processes that information, it sends out information based off of what came in. We have to have some kind of motor output. Could be your muscles, could be your stomach, your bladder, all your organs, your heart, even. They all have to have some kind of response, some output, depending on what the brain is, what signals the brain is sending. Like, say you're thirsty, you see a glass of water, you see that water, that's sensory input going into your brain, seeing that water. The integration center, of course, is your brain. That's where your brain says, oh, okay, let's go pick up that cup of water. And your brain sends the information out to your muscles that says, hey, pick up the cup of water. Let's drink some water here. So, as I've mentioned, we have different nervous systems. We have a central nervous system, that is your brain and your spinal cord. It allows for different sensory inputs, like, say, it's cold. So, that information for cold goes into your brain. And depending on where you're at, your response could be goosebumps. It could be shivering to help warm the body back up. Peripheral nervous system. Peripheral nervous system are all the nerves outside of that brain and your spinal cord. So we have spinal nerves. These are nerves that go out or come in to your nervous system. We have cranial nerves. These are nerves that come right off the brain, and they usually go, most of them go somewhere in the head. We'll get some in the neck. We have one that goes all the way down and into our abdomen to do some different kinds of work within there. But those are the two kinds of nerves that we have. So this picture just shows our central nervous system is that brain and that spinal cord, while the peripheral nervous system could be those cranial nerves, those spinal nerves. And we also have what are called ganglia. These ganglia here, we'll go in more detail about them later, but what they hold is they hold the cell bodies for those spinal nerves. So we'll go into detail about what those do when we get to that point. So within our peripheral nervous system, it gets split out even further. Because within our peripheral nervous system, we have that sensory or afferent division. Remember all the way back to chapter 1? Afferent is what sends information into the brain. So we have somatic sensory fibers. Somatic, we said deal with muscles when we were in the muscle chapter, but it also deals with skin, muscles, and joints. It sends information into the brain based off of what your skin feels, what your muscles are doing, how your joints are moving. All of that information is always constantly going into your brain, whether you know it or not. Then we have a visceral sensory fibers. These come from your visceral organs, like your stomach, your small intestines, your large intestines, your bladder, your pancreas, your liver, your spleen, your heart, your lungs. These are all visceral organs that send information up to your brain. And our peripheral nervous system has motor efferent. Remember, efferent goes back out. So an efferent nerve takes that information that the sensory or afferent nerves sent to the brain will send information back out on these motor efferent neurons for something to happen, for you to move, goosebumps to happen if you're cold, sweating if you're hot. We have a somatic nervous system and an autonomic nervous system as well. We will go into those a little bit here, and then we'll go into more detail with them later. So that somatic nervous system, somatic is nerves, there's muscles. So they're the nerves that go to the skeletal muscles. It is considered the voluntary nervous system because we can voluntarily tell our muscles to do something. Even though most of it's done unconsciously, we can still tell our muscles to do different things. We have our autonomic nervous system. That has to do with our sympathetic and parasympathetic, that fight or flight or rest and digest. I'm sure you've all heard those terms. That's all part of our autonomic nervous system. It is involuntary. We don't have control over that fight or flight function. We don't really have control over that rest and digest type function either, because they work opposite each other. So here is our central nervous system and our peripheral nervous system. We show everything that makes up the central nervous system. Then we have everything here that makes up the peripheral nervous system. We have that sensory afferent into the brain, motor efferent, the responses that come back out. They can be somatic when the information comes out, so it deals with the skeletal muscles. Or it could be an autonomic response that deals with most of your visceral organs. We have that sympathetic or that parasympathetic. all of these different divisions within that peripheral nervous system. So let's look at the histology of our nervous system. Our nervous tissue is either neuroglia or glial cells. They're cells that surround and wrap delicate neurons. Then we have neurons. These cells transmit electrical signals. That's how they communicate with each other, is through electricity. I know I mentioned in the muscle chapter, but neurons are excitable cells, just like muscle tissue is excitable has excitable cells. Those are the only two really excitable cells in our body: nervous tissue, muscle tissue. So the different kinds of neuroglial cells, these are cells that help support our nervous system. These ones are specific to the central nervous system, because there are some that are specific to the peripheral nervous system, but these are specific to the central. So we have our astrocytes, microglial, ependymal cells, and oligodendrocytes. So let's look at what all these do. Our astrocytes. Our astrocytes are very branched. They hold on to neurons, or synaptic endings. They hold on to capillaries, but they kind of support those neurons. They exchange between capillaries and neurons. They help young neurons get where they're supposed to go, lead the direction. They help with that chemical environment, help with everything around that neuron. They respond to nerve impulses and neurotransmitters. So those action potentials we talked about when we talked about muscles, neurotransmitters, we talked about those as well. How some of the chemicals that are used to communicate between cells. They kind of help influence how our nerves work, and they participate in processing information as it comes into the brain. We need all of that. So here's this astrocyte. You can see how this astrocyte is wrapped around the capillaries. It's got some projections wrapped around this yellow neuron. These astrocytes just kind of reach out and wrap around everything they can, because they're kind of helping to make sure everything is working the way it should, kind of like osteocytes in our bones, kind of like chondrocytes in our cartilage. They all kind of they work kind of the same. They're kind of help monitor, make sure everything's working the way it should be working. Microglial cells. These have a more thorny process. They still touch and monitor neurons, just like the astrocytes. But microglial cells will find these injured neurons. They go in when there is an injured neuron, they clean up everything, because they can phagocytize, which means they ingest microorganisms and neuronal debris. So if a nerve breaks, something happens to it, it gets damaged in some way, these microglial cells are going to be the ones that come in, make sure nothing happened, there's no viruses, bacteria, any kind of little microorganism in the area. If there is, they're going to clean it up. They're also, if there's any kind of debris from like cells that broke off and kind of just laying around, they're going to get rid of that as well. They're cleaning up the environment around these injured neurons. So here's these microglial cells. They have all these little finger prickly, they almost look like thorns coming off on a rose bush. That's kind of what it looks like. But they're touching these neurons around them, making sure everything is okay, nothing is injured, everything's functioning the way it should. That is the role of the microglial cells. Ependymal cells. They're kind of have different shapes. Squamous and columnar. Sometimes they're ciliated, but when they are ciliated, you know that cerebral spinal fluid, they're going to help move that cerebral spinal fluid around. Our cerebral spinal fluid kind of flows through our nervous system. It doesn't just sit in one area. Once it's there, it doesn't really move. It does. It flows around our system, flows down our spinal cord, flows back up. It's always kind of moving around. But it lines the cavities of the brain and that spinal cord. I just said it helps to move this stuff around, so it's constantly moving in different directions. It gives us a permeable barrier between the cerebral spinal fluid and any other tissues in that cerebral spinal fluid. So here's these ependymal cells. You can see all the cilia coming up off of them. They're helping to beat and move the cerebral spinal fluid so it's not going all over the place in many different directions. Its job is to move the fluid around. That is its main job. Oligodendrocytes. Oligodendrocytes in the central nervous system wrap around nerve fibers. They make what is called a myelin sheath in these thicker nerves. So here's 3 different nerve fibers. You can kind of see them going through here, going through here, and going through here. They wrap around, these oligodendrocytes are wrapping around these different axons of these nerves in the central nervous system. It kind of helps to insulate them. It helps to protect these neurons so they're less likely to get damaged. And it is what they're making is what's called a myelin sheath that wraps around each of these neurons. We want to protect our neurons as best as we can, because by protecting our neurons, it keeps our nervous system working correctly. So that is the job of these oligodendrocytes in the central nervous system. There are different ones that do the same job in the peripheral nervous system. So oligodendrocytes are specific to the central nervous system. Now we can look at what cells help our nervous system in the peripheral nervous system. We have what are called satellite cells. Their functions are pretty similar to astrocytes. They surround the neuron cell body and kind of monitor what's going on. Then we have Schwann cells. Schwann cells do the same job as oligodendrocytes, but in the peripheral nervous system, because they are going to form those myelin sheaths that wrap around the axons of nerves. So here are these satellite cells. They're literally wrapped themselves around the cell body. Then we have all these Schwann cells in blue. So here's a Schwann cell. Here's a Schwann cell. All these Schwann cells, they're helping to protect this neuron or the axons of this neuron. It's helping to keep this neuron from getting damaged in any shape or form. So what exactly are neurons? We've talked about them a little bit, but neurons, they're what conduct impulses. They're going to feel something going on. They're going to send what they're feeling to the nervous system, to your brain. Your brain is made up of thousands of neurons, which will then send information back out to respond to whatever they are feeling or sensing here. A single neuron will last your entire life. They are amitotic. They can divide by simple cleavage of the nucleus. It's very rare for this to happen, but it can happen. They require a lot of oxygen and glucose to function. They need a lot of ATP to send information because their nerves are working all the time. Even though you're not acknowledging everything that your nerves are sending to your brain is what then decides whether or not you should be aware of what is actually going on. They have a cell body and they have one or more processes. The cell body of the neuron is called a perikaryon or a soma. That cell body, it makes proteins, membranes, it makes different chemicals. Like I talked about when we talked about muscles, that acetylcholine, part of that acetylcholine is made in the cell body, and then it is sent all the way down that long axon out to the nerve terminal where the nerve communicates with that muscle. Well, nerves are gonna make all kinds of different proteins just depending on what kind of neuron it is, where they're going. They have a rough endoplasmic reticulum. They have a very spherical nucleus or nucleolus with a nucleolus around it. The nuclei is a cluster of neuron cell bodies in the peripheral nervous system, while a ganglia, I had pointed it out towards the beginning when I mentioned we'll talk about them later. A ganglia houses the neuron cell bodies of the peripheral nervous system. And most of them are just outside that spinal cord. So our neuron processes they're kind of arm-like. Some of them are really long, other ones are not as long, but they come off of that cell body. In our central nervous system, we have neuron cell bodies and their processes all within the central nervous system. But in the peripheral nervous system, we usually just get the neuron processes because that is what we would see coming off of the different cell bodies. We have tracts. That is where lots of neurons There are lots of the processes off the neurons in the central nervous system. We have a lot of tracks on our spinal cord. We'll look at some of those when we go to the central nervous system. Of course, we have nerves. Those are the neuron processes in the peripheral nervous system. We have dendrites and an axon. An axon is usually what we see if you do find any nerves in the cadavers. Those are usually the axons in that peripheral nervous system. So here is a nerve. We have the cell body right here. The little processes here that come off the cell body. They have those little thorny-like processes that come off. Those are called dendrites. While this long process that comes off the cell body, this is what is called that axon. This is what's either receiving information from outside world or from whatever peripheral organ or muscle, or it is sending information or it's receiving information to tell you to do something. Move your arm, move your leg, do whatever. This is the part that we would see, that you can see in your peripheral nervous system, is that these nerves go to muscles. So what are these dendrites? There's usually hundreds of these dendrites off of a single cell body. And they are the receptive region of the neuron. So information comes into the neuron through these dendrites. They usually come as some kind of graded potentials. We'll go into what graded potentials are. But in short, they are short distance signals. They could have very specialized functions. They could collect very specialized information. That axon starts at that axon hillock, which, coming off of that cell body, there's that little cone-shaped part that comes down. This is the axon hillock. But that is that nerve, that long structure that goes out to talk to muscles, that goes out to talk to your organs. They can be over a meter long. I think I mentioned this in the muscles chapter, but think about it. The nerves coming out of your spine going down to your big toe. That's a long ways to go, and it's one single axon that gets there. This is what we usually call those nerve fibers. Sometimes they'll branch, like in muscles when we talked about in skeletal muscles, each of those different skeletal muscles that we have. Those nerves kind of branch. One will go here, one will go to this muscle fiber. They branch off and they go to every muscle fiber in that motor unit. Well, that's how these nerves will branch, and they're called axon collaterals. They tend to branch a lot at their end. As many as 10,000 branches can go off to all those different muscle fibers per se. And at the end of that neuron is called that axon terminal. We looked at that when that part that comes out and communicates with that muscle, this portion right here is that axon terminal. So what does that axon actually do for the nerve? Well, it's that conducting region. That is where information is sent to or from. It'll generate nerve impulses or action potentials. It sends information along that axon or axolemma, which is the cell membrane, all the way up to that, all the way down, sorry, to that axon terminal. That is where those neurotransmitters get released, just like what we talked about with muscles. We have our axon terminal coming out. I'm showing with our muscles because that's what we've already looked at. So this is where neurotransmitters get released out into. Those neurotransmitters can excite something, like they can tell a muscle to contract, or they can inhibit something. Nerves can multitask much better than we can when trying to do multiple things at once. Our nerves can have many different conversations at the same time. I think I'd get way too lost if that was happening around me, but your nerves can handle it. They're talking about many different things at the same time. They send their information to those cell bodies, which then send their information onto the brain. Their cell bodies are what make new proteins for them. Those proteins help to make their different neurotransmitters. But if your axon gets cut or damaged, that nerve breaks down pretty quickly because our nerves aren't that good at repairing themselves. They try, but they're not that good at it. So they use different internal transport mechanisms to send information along. We have anterograde movement and retrograde movement. Anterograde goes away. Retrograde goes towards. So our mitochondria will send stuff away. The cytoskeletal elements of the membrane components, those go away from the cell body. Our enzymes, those need to go down to that axon terminal. But if something needs to be degraded, be broken down, because it's no longer of use, it gets sent back to that cell body to be broken down and to make new signal molecules, like those neurotransmitters. Once those neurotransmitters get used, there's different enzymes that break them apart. Once they're broken apart, they can be taken back up into that neuron where they're sent back to the cell body to be remade again so they can be used again. Viruses. Our nerves can catch viruses, send it to the cell body and be destroyed. Same with different bacterial toxins. They're trying to help keep us healthy all at the same time. But some of those viruses and bacterial toxins that use that retrograde axonal transport, so in stuff that goes back to the cell body, some of those viruses and bacterial toxins use that to their advantage. Polio, that's a big one. Rabies, herpes, simplex viruses, and tetanus toxins. Those use that retrograde transport system to take over some of these neurons. So now that the scientists have figured out that we can use this retrograde axonal transport, that these viruses and toxins do this, they're trying to use this concept to treat different genetic diseases that may affect nerves. Because viruses like to get into our nervous system or get into our cells. They use our cells' information to replicate themselves. And then once they're finished with that, they break out of that cell and they go on to the next one and they do it again until they get caught by our immune system. So they're using that idea, using a virus, because a virus is either RNA or DNA. They can send that information into our system to try to suppress defective genes. So they're trying to do that. They're getting into neurons and going back to the cell body and fixing some of the problems by using our cell's own information to do so. So both our central nervous system and our peripheral nervous system has some kind of myelin sheath, whether it's Schwann cells or oligodendrocytes. Oligodendrocytes are the central nervous system, Schwann cells are the peripheral nervous system. But they have this protein called myelin. It's actually a protein and a lipid together. But of course, it's going to protect that axon. We want that. It also helps to insulate that axon. It works really cool to help speed up nerve transmission. So I have my axon here. I'll give you a quick demonstration of this. It wraps around this neuron, but you notice there's all these little gaps between cells. How this works, because we talked about with muscles, when the muscle receives information, that information gets sent along that muscle fiber in both directions wherever that nerve is attached. Well, neurons do the same thing. They send information down or back up their axon. And what's cool is where these myelinated sections are, information jumps over that myelination. That's what helps to speed things up. It just jumps, 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 jumps. But we'll go into more detail about exactly what's happening, but that's just kind of the general idea on how it speeds up that transmission. We have myelinated fibers around most of our long or large diameter axons. But we have some non-myelinated sheath fibers as well. But when they do not have that myelin sheath, they're a lot slower at sending information. So our neurons transmit information down different neurons depending on how fast something needs to be known to our brain versus, does our brain need to know this right this second or not? So as I said, our peripheral nervous system has Schwann cells. Just like our central nervous system has oligodendrocytes. I like how your book says that they wrap around the axons like a jelly roll. If it helps with the visual of what it looks like, then awesome. One cell forms one segment. So think of how many millions of cells make up of Schwann cells we have in our body. Quite a few. We have a perinuclear cytoplasm in these Schwann cells. Of course, they have a nucleus, and they have to have cytoplasm. We don't really have channels or carriers on our Schwann cells because their main job is an electrical insulator. So here's how myelination happens. When a Schwann cell will envelop around that axon, then it starts wrapping around, and it wraps around some more until we have this full myelin sheath around this entire axon, helping to insulate it, helping to also protect it. So here is a cross section of an axon. You can see just how much myelin sheath there is. You can really see at the bottom all the different layers. You don't see it as well at the top, but you can see it really well at the bottom. Then that axon is the part in the middle. That is that neuron. The gaps between the Schwann cells, they are called myelin sheath gaps now. I had learned them of nodes of Ranvier, but they are now called myelin sheath gaps. Under non-myelinated fibers, those thin fibers are not wrapped in myelin. They're still surrounded by Schwann cells, but they're not coiled. Like in this picture, you see all this coiling happening. That's a lot of coiling. They don't do that on these non-myelinated fibers. So on the non-myelinated cells, we tend to have a lot more. Those Schwann cells tend to surround many different fibers instead of just one on the myelinated cells. So here's this neuron again. We have our cell body. We have all our different dendrites through here. We have our axon hillock through here. That is that cone shaped. Then we have our axon that goes along this way. We have lots of these Schwann cells wrapped around. Then we have all these different gaps, myelin sheath gaps between each of these Schwann cells down that entire axon. So how is the myelin sheath different in that central nervous system? Well, of course, there's different cells, but it's also not the whole cell, because these oligodendrocytes can wrap up to 60 different axons at once. So these Schwann cells on the myelinated axons, they wrap around the same axon. The entire cell is in one spot. But the oligodendrocytes wrap around up to 60 axons. They have lots of those little projections that just wrap around them. Still, the thinnest fibers are unmyelinated, but they're still covered with those neuroglial cells. We have what is called white matter and gray matter in our central nervous system. Where we have white matter in that brain and spinal cord, those are myelinated fibers. Gray matter is either cell bodies or non-myelinated fibers. We'll point that out again when we get to the central nervous system, and we'll be looking into this in a lot more detail. So let's look at the different kinds of neurons. We have 3 different kinds of neurons just based off of how their processes are. We have multipolar. Our multipolar neurons have 3 or more processes. So they have one axon and other dendrites. This is the most common. Multipolar, you're going to find these around majority of the nerves in your body. Bipolar, one axon, one dendrite. Your retina in your eye, in your nose, that olfactory mucosa in your nose, those are bipolar. Unipolar, kind of like a T-like process. There's 2 different axons, or pseudo-unipolar. We have a peripheral process that's usually sensory, and a proximal process, that's what goes into our central nervous system. So let's look at these. This first one here, this is our multipolar. You can see you have your cell body, you have a big long axon, and then you have lots of these dendrites coming off. While the bipolar, you have a you have a dendrite and then an axon, the cell body in the middle. Unipolar or pseudo-unipolar, we have our cell body in the middle. We had a peripheral process and then a central process. So that peripheral process is usually some kind of sensory. Central is going towards the central nervous system. So here is the different parts of what these neurons do. We are looking back at that multipolar. The receptive region, that is the region that receives information. The conducting zone, that's that axon. Then we have the secretory region. That is what releases neurotransmitters to communicate with other different types of cells or even different neurons. We look at this one, we have a really big receptive region. That's where all the information gets sent in. Conducting, that is where we're sending information out. Secretory, that is where we are sending out neurotransmitters. Receptive region, that is sensing sensory input. Transmitting region, secretory region. And where these are found, multipolar, most abundant, mostly in the central nervous system. Here's some different structural characteristics. This one looks like it has a lot of hair. This one looks like roots of a plant. This one kind of looks like a tree. Got roots at the bottom and branches at the top. I can make these look like anything you want me to. This last one, the unipolar, pseudo-unipolar. We've got receptive ending, cell body. This is going towards our central nervous system. So these neurons, there's different neurons for different things around our body. So now we can look at the functional classification of neurons. Yes. Not only are they're different shapes based off of what they do, there are different types of neurons based off of what they do. A sensory neuron. A sensory neuron, of course, is going to get sensory information. Like when someone touches you, hot, cold, sharp, dull, touch, vibration, all of that different sensory information that is going to be sent from your periphery, like your skin, your muscles, back to your central nervous system. They're mostly unipolar. Their cell bodies are usually located in those ganglia. Remember that picture from the beginning that had all those they have these little bulge things up here? There's tons of cell bodies from the nerves that are coming out of the spinal cord. Their cell bodies are located in here. We have motor neurons. These are usually multipolar. Of course, motor, they're going to send information back out on what needs to happen. Shiver, goosebumps, sweating, run away. Their cell bodies are in that central nervous system, except some of the autonomic neurons. Those autonomic neurons are neurons that are going to say your organ systems, your glands. Those are more autonomic type of neurons versus somatic, which go to skeletal muscle. And an interneuron, they're the ones that go between the motor and the sensory. They're almost all within that central nervous system. Most of your neurons are interneurons because we have that nerve that feels something. It goes all the way into the spinal cord. Then it connects with an interneuron. That'll go up to the brain. The brain will send information down to an interneuron. That'll go out to do something. So here's the same kind of neurons. We have our multipolar, bipolar, and unipolar. And what some of them do. Multipolar, motor neurons. Bipolar, some kind of sensory neuron. Unipolar, those are also some kind of sensory neuron, like in your skin when somebody touches you, taps you on the shoulder. That's one of these unipolar neurons working, so you can feel somebody touch you. We talked about it a little bit with muscles, but all cells have a resting membrane potential. It's usually between -70 to -90. And like we've talked about with muscles, neurons can also rapidly change their resting membrane potential because they are highly excitable. They like to be communicated with. They like to talk to people, other neurons, and they get very excited when they're talking to other neurons. So it's something that nerves like to do. Just like magnets, opposite charges are attracted to each other. So positive and negative, they are attracted to each other. Why do we need so much oxygen and glucose in our nervous system? Because those positive and negative charges, they like to be right next to each other. So it takes energy to keep space between them. Have you ever tried to hold two really strong magnets, positive charge and negative charged sides, close together, but not let them touch? It takes a little bit of work to keep them there because they're trying to pull each other together. So we're going to go into these basic principles of electricity. We have what is called a voltage. A voltage is the potential energy generated by the separation of charges. It's volts or millivolts. Can be a potential difference or just a potential. So the greater the charge difference, the higher the voltage. Current. That's that flow of ions moving between points. Ions have that positive or negative charge, like sodium, potassium, calcium. They all have some kind of charge. That's an electrical charge. So that current is that flow of electrical ion charges or ions between those points. We can use that flow to do work, like have neurons communicate with each other. And that flow is dependent on voltage and resistance. But what is resistance? That slows down or and hinders the charge flow. We have an insulator. That insulator has high electrical resistance, while the conductor has low electrical resistance. So let's look at Ohm's law. What Ohm's law says is our current, which is I, equal to voltage divided by resistance. So our current is proportional to voltage. So the greater the voltage, the greater the current. The current is inversely proportional to resistance. So the greater the resistance, the smaller the current. Let's look at how ion channels work. Our nervous system uses a lot of ion channels to communicate with each other. Those ion channels allow different ions, of course, through. We may have potassium channels. Those potassium channels are only going to allow potassium through. We have sodium channels that only allow sodium through, and calcium channels that only allow calcium through. Same with chloride. We have leaky channels. They're these non-gated channels, so they're always open. Then we have gated channels. Those chemically gated, we looked at those in muscles when we looked at that channel that allowed neurotransmitters to connect. And that's what opened that channel. We have voltage gated channels. Those voltage gated channels are going to open depending on the membrane charge. Like if our membrane charge gets up too high, too positive, it's going to open certain voltage gated channels to get positive ions outside of its cell. We have mechanically gated. So they're going to open depending on, say, some kind of touch. So as I've said, chemically gated, they have ligands that attach, usually neurotransmitters. Voltage gated, they open and close depending on the charge of that membrane. And mechanically gated, they open due to like some kind of physical deformation of receptors. Like if someone touches you hard enough, it causes these mechanically gated channels to become deformed and open. So here's examples. Are chemically gated, we need some kind of neurotransmitter to allow the channel to open. We have those voltage gated. It's only going to open when that charge along that membrane becomes a certain charge. So as soon as that gate opens, ions flow in or out extremely quickly. They like to go from high concentration to low concentration. So there's usually more sodium outside of a cell and more potassium inside of a cell. So sodium is going to flow in, and that's why potassium flows out, because they're going from an area of high concentration to that area of lower concentration. That electrochemical gradient. That is electrical and chemical gradients combined. So things are going to open depending on what is happening. When ions are flowing, it creates an electrical current and a voltage change across that membrane. That's why voltage is equal to current times resistance. It's rearranging Ohm's law to talk about how everything is related to each other. So we have what is called a voltmeter. It can actually work to kind of measure neurons and their conducting of information. So I've said our resting membrane potential is usually around -70 in a neuron, but it can be -40 to -90. Our resting membrane potential is said to be polarized. And then potentials can be generated by the ion composition in the intracellular and extracellular fluid, like I've said, sodium and potassium. That plasma membrane permeability is another one. How permeable is that membrane to sodium versus chloride? So here's a voltmeter. This voltmeter is reading and showing us that inside of this axon of this neuron, that charge is currently at negative 70 millivolts inside the cell. Outside of that axon, the charge is more positive. Shows the charge difference between the two. And I've even said it, higher concentration of sodium in the extracellular fluid. Intracellular fluid has more potassium. So we're able to generate this resting membrane potential because of the differences in potassium and sodium concentrations, and the differences in the permeability of the plasma membrane to these ions. So how easily can these ions get into and out of each neuron? As we talked about when we talked about cells back in, I think, chapter 3, we said that most plasma membranes, larger ions cannot get through. They're slightly permeable to sodium, but that's because we have leaky channels that allow sodium in and out. But our plasma membrane is a lot more permeable to potassium than sodium because we have all these leaky channels for potassium. And there's more potassium inside the cell than out, so potassium's going to diffuse out of that cell. We're also pretty permeable to chloride. Chloride is one of those negative charges, negatively charged ions. So in general, potassium usually goes out of the cell. More potassium goes out of the cell than sodium that comes in through those leaky channels. But that's what makes inside of that cell more negative. And that's what establishes our resting membrane potential, is having more negative inside, more positive outside. We have our sodium potassium pump. And that's what helps to keep that resting membrane potential when there's nothing going on between those neurons. We need to keep more potassium out of our cell, more sodium out of our cells and more potassium in our cells. So these sodium potassium pumps, what they do, here's an ion channel. They are going to they're going to pump 3 sodiums out for every 2 potassiums in. So 3 positives out for 2 positives in. That's how it keeps that more negative charge. Because more positive is going out than what's the negative than the positive that's coming in. Let me say that again. There's more positive going out of the cell than there is positive coming in the cell. And that's what helps to keep that negative charge in that membrane. So here is showing how we pump 3 sodiums out for every 2 potassiums that come in. We also have these leaky channels. These channels that only allow potassium or sodium in. Then we have our pumps that pump against to help maintain that concentration gradient. So what changes that membrane potential? What changes the possibility of our cell going from negative 70 millivolts up to, say, positive 30 millivolts? Well, as these ions travel back and forth across that membrane. How permeable is that membrane to these ions? That's all going to make a difference. We have graded potentials and we have action potentials. Graded potentials go short distances. Action potentials go down those long axons, long distances. So these membrane potentials is how we receive, integrate, and send out information. So depolarization. I know I had mentioned this with muscles. So if our resting membrane potential is right here. When that action potential that comes in, our cell becomes more positive. That's depolarization. Because our cell is becoming less negative than our resting membrane potential. Hyperpolarization. This is where we repolarize, but we went back down below resting membrane potential. That is when we're more negative than resting membrane potential. That is our hyperpolarization. So here is the same thing that I just drew. Depolarization is where we become less negative, while hyperpolarization was where we become more negative. So here is where we're depolarizing. We became less negative. Here's where we hyperpolarized. We became more negative. Graded potentials are pretty short-lived. They don't change a lot in that membrane potential, but the stronger the stimulus, the more voltage changes and the further the current flows. We need some kind of stimulus that opens that gate, like a strong enough touch that changes the shape of that gate to open it. We usually get some kind of depolarization. Sometimes we'll get a hyperpolarization out of a graded potential. We have a receptor potential. Those are usually in some kind of sensory neuron. Or we have a postsynaptic potential. These are neuron graded potentials. So let's look. We have this stimulus that comes in and touches this axon at this specific spot. This is going to stimulate a potential here. The red is the area that depolarizes when we get touched. That causes this specific area to depolarize first. But once those ion channels open, that depolarization is going to spread like this. How it started here, but now it's traveled. It's traveled here, it's traveled here. Here it's traveled, it's keep traveling out. But as it keeps traveling out, because it's going to keep sending information down this way, keep sending information down this way, where everything started from is going to start to repolarize as the depolarization spreads out along that axon. But usually dissipates and it decays. It doesn't usually send information very far. So here's how it shows. This is where that axon was originally depolarized. Something touched it. Something triggered it to depolarize. It sent its information out, both directions, but it didn't last long. It kind of died off. Because of that leaky plasma membrane. It's going to keep things from working efficiently sometimes. Action potentials, on the other hand, that information comes in and it can have, and information gets sent out. Action potentials only happen in muscle cells and those axons of neurons. We also know that action potentials do not decay. That information keeps getting sent along. That's why it's called a nerve impulse. Because once it starts, it keeps going. So here is what happens. We've looked at this a little bit with muscles. We're at a resting membrane potential down here at -70. Oh, something came in and touched us. It opened up these sodium channels. Sodium channels, sodium comes flying into the neuron. Oh, our cell got up to positive 30 millivolts. Uh-oh. Sodium channels closed. Potassium channels open. Potassium comes flying out of that neuron as fast as it can, and it repolarizes. Oh, too much potassium flew out. We're now hyperpolarized. Those potassium channels close. Those leaky channels and those sodium potassium pumps will pump ions back across until we get back to that resting state of -70. So as I've talked through that, what happens first? Well, our sodium channels. Something touched us, which causes a response. When that happens, it opens up these sodium channels. These sodium channels sodium comes flying into that neuron. Up, that is when we hit positive 30 all the way up here. Well, our sodium channels need to close, but they're not super good at it. So they have this inactivation gate, that ball on a chain that I talked about with muscles. Well, they've got them in nerves too. That closes things up so sodium can't come in anymore. And once that inactivation gate comes into play, that inactivation gate causes this channel to not be effective. We cannot use it until the channel gets back into its original closed state. It has to go shift back to the inactivation gate off with the activation gate closed before we can receive another action potential. Our potassium channels also play a huge role because when our cell gets depolarized, we need to repolarize it. These potassium channels are opened, as it says, by depolarization. Our cell gets too positive. We don't want our cell to be too positive. So our cell's response is to open potassium channels and allow potassium to flow out of that cell until the cell becomes negative again. Our cells don't like to be positive. They like to be negative. So that is how our potassium channels kind of play into all of this. So let's kind of look at how this whole thing happens. I am going to open a blank slide again. So we have a neuron here. And it is communicating with another neuron here. An action potential comes down the neuron. Inside my neuron here, I've got all these vesicles with neurotransmitter. What causes these neurotransmitters to be released? As this nerve impulse or this action potential comes down, it's going to cause calcium channel gates to open. Calcium channel gates open in this nerve terminal. So calcium comes into that nerve terminal. That is going to cause neurotransmitter to be released out into that synaptic cleft, the space between the two neurons. What happens next? Well, we've got these channels here. These channels here, just like on a muscle cell, we have these ligand gates. So that neurotransmitter is going to attach. That causes these gates to actually open. I drew them open, but these neurotransmitter connecting is what causes them to technically open. Once they open, sodium comes rushing into the cell. So let's look at what's happening here on a graph. Got my resting membrane potential. It is at negative 70 millivolts. Something comes in and stimulates. Oh, causes that neurotransmitter to be released. Neurotransmitter connects to the ligands of the next neuron, which causes the sodium channels to open. Oh, we hit positive 30 millivolts. That is going to cause these neurotransmitters to get broken off, get degraded, and cause these sodium channels to close. It's also going to cause potassium channels to open. Potassium is now leaving the cell. As potassium leaves the cell, we're repolarizing. We lost a lot of potassium. Remember those sodium channels I showed you that has that ball and chain? First thing that happens with those sodium channels on that ball and chain, that inactivation gate comes up and closes this first. And then it'll go to a closed state here. It needs to go back to from was open, took that inactivation gate, closed it. It needs to switch back to being completely closed before the next action potential can be sent through. But we let so much potassium out. Now our potassium gates need to close. Now what do? We've let too much out. Our cell is too negative. Well, remember those pumps. We're going to pump free sodium's out and 2 potassium's in. While that is happening, we are getting ourselves back to our resting membrane potential. It's happening at every one of these. So we're getting 3 sodiums out, 2 potassiums back in. Now, what is happening while we're waiting for these gates here to switch back to their resting state? That is where we will continue looking at this. So this just goes through the steps of what is happening. I wrote it all out for you. So you also have it here on what is happening, even to the hyperpolarization. So here's all the steps, everything that is happening. With everything closed, an action potential comes in and opens the sodium gates. That inactivation gate, potassium leaves. Hyperpolarization, that sodium gate is going back to its resting, and we're pumping stuff in and out to get it back to our regular potential. So repolarization resets electrical conditions. As I talked about with the picture I drew, we're getting the potassium back out because we're trying to get back down to that negative 70 millivolts. It resets that electrical condition. We're trying to get back to that negative 70, not ionic conditions. That is where our pumps come into play. Our pumps are what cause us to get back to our ionic conditions with more potassium in the cell, more sodium outside of the cell. So I've kind of talked about some of this a little bit, but now we're going to go into detail. It says not all depolarization events produce action potentials. Well, what does that mean? Remember this chart I drew with our resting membrane potential here? And we get this depolarization, this repolarization, hyperpolarization. Uh-oh. Well, let's kind of look into that. So for an axon to fire, or to let those gates open and close the right way, first thing that must happen is we must reach a threshold voltage. That's what may cause an action potential to occur. So let's look at this a little more. Little bit bigger here. So I have my resting membrane potential. Then I have my threshold potential. Resting membrane potential is negative 70. Threshold potential, let's say it's negative 50. Because it's usually 15 to 20 millivolts. So here's my resting membrane potential. Here is my threshold potential. Let's say there's somebody comes up and touches you on your back. They tap your shoulder, but they tap it so light. They touch, it changes those gates a little bit. Nope, did not send. Did not reach our threshold potential, so nothing happened. They touch you a little bit harder. Change the shape of that gate just a little bit, got a little bit of sodium in to that cell, but we didn't reach threshold potential for an action potential to start. They come in and they tap even harder. They touched you hard enough that you felt something. Well, that's where we reached that threshold potential. So now we depolarize the cell. It'll repolarize. Oops, and it'll even hyperpolarize. This is what this is talking about here. So if something comes in and touches you or something comes in and happens, it needs to hit it all the way. It needs to be something that hits that threshold potential for that action potential to happen for us to feel it. So when we get to that threshold potential, again, it's 15 to 20 millivolts above resting membrane potential. When we're at threshold, our sodium permeability increases. So sodium, that's where sodium comes flying into the cell. Potassium comes flying out of the cell. And it's a positive feedback cycle. And for an action potential to happen, it's either all or none. we either set off that action potential or we don't. That is where what was happening right here, this is where no action potential happened because we didn't reach threshold potential. Here, we reached threshold potential, so we got an action potential. They're not graded based on how hard somebody touches you. It's graded on being able to reach that threshold potential to set off that action potential or not. Because we didn't even feel these first two because we never reached threshold potential. That is what that means. Propagation. The propagation is that action potential being sent down the axon. That information that we just received from somebody touching you gets sent all from that nerve ending that felt that touch all the way back to that cell body. That's what propagation is. It needs to send all that information to that cell body that goes to the spinal cord, that goes up to the brain, so the brain can let us know, hey, somebody touched you. So that sodium that comes flying into the cells, they go through those voltage gates. They go through those ligand gates. But that's what causes the cell to depolarize, or that axon or neuron to depolarize. Once that one area depolarizes, the next area depolarizes. Then the next area depolarizes. That action potential is self-propagating, so it's going to continue to travel down that cell, that axon. That's where I said, remember those axons, the non-myelinated ones? That information gets traveled across that axon. Those myelinated axons. So we have our myelination through here. Now I said that is where information jumps over that myelination from one to the next, to the next, to the next. And because those sodium channels, they have that inactivation gate that's still up, we cannot receive a new action potential until those sodium gates are back to their resting potential before the next action potential can come in. Nothing can happen until everything is back to its resting state. So here is where it's showing. We just had a nerve impulse and it's traveling down the axon. You can see how it's still traveling down the axon and down the axon. It keeps traveling. And we start negative on the inside, positive on the outside. Well, they switch, more negative on the outside, positive on the inside, and then the channels along the way pump that positive and the negative back until we're back to our resting state. So as I said, it doesn't matter how hard somebody touches you, all action potentials are the same. The stimulus intensity does not make a difference. The action potential, the information being sent along is still the same. But our brain is really smart. because they can tell a weak stimulus from a strong one just by the frequency of impulses. How fast are these impulses coming at you, at the brain? The faster the impulses are coming, the stronger the frequency or the stimulus. The slower the action potentials are coming, the weaker the stimulus. So here's just an example that shows. This first one, our threshold is about here. This first stimulus, that is called a sub-threshold stimulus because it does not reach threshold to stimulate an action potential. This was strong enough to stimulate an action potential. That was 4 stimuli, 4 action potentials sent. Boom, 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 one after another. Well, look at this. This is a whole lot more. That must have been a really big stimulus because there were several action potentials generated from it. Now let's talk about what happens when we're still waiting for that sodium gate with the inactivation gate blocking that sodium channel, so we cannot start a new action potential because it's not back to resting yet. That period is called a refractory period because we cannot send off another action potential until that sodium channel is back to resting. That is when we have this activation gate in place. We're waiting. We need this channel to close with the inactivation gate off. This is what we're waiting for before the next action potential can go through. Part of this refractory period, those sodium channels are still open. When those sodium channels are still open, we can't send out another action potential. When these sodium channels are blocked by the inactivation gate, we cannot send out another action potential. So that's absolute refractory because until they're settled back to their resting state right here, we can absolutely not have another action potential because this inactivation gate is blocking it. That is where it's an all or none event. Because an action potential can come down and hit that next neuron, but until that inactivation gate is back to resting state, nothing's going to happen. That is how our nerves stay a one-way transmission. We have what is called a relative refractory channel or period. Relative refractory, it's right after that absolute refractory time. But majority of the sodium channels are back to the resting state by now. So we're at this part during relative refractory. But we're still repolarizing that cell because some of those potassium channels are still open. So what that does, I have my resting membrane potential. I have my threshold potential. But since we still have some potassium gates open, we're still repolarizing, we have an elevated threshold potential. So for the next stimulus to get sent through, it has to stimulate a whole lot higher to send off that action potential. If it stimulates, but say it only stimulates up to here. Well, stimulating up to here, nope, we didn't hit that elevated threshold potential, so nothing happened. So we needed very strong stimulus to cause another action potential to happen at this point. Your book says to think of a disobedient, quote unquote, refractory dog. If you call your dog during an absolute refractory time, they're not going to come. They'll never come when they call their name during that absolute refractory. Relative refractory, if you call their name loud enough, they may come. It just depends on how loud you call their name, if they know their name. So here's our absolute refractory versus relative refractory period. That absolute refractory, as you can see, this is where our sodium channels are open. This is where our sodium channels are going back to their resting state because the sodium channels tend to close up here. So they need time to go back to that resting state before another action potential can come in and trigger them. But then since we're not back to our resting state, it needs to be much higher of a stimulus to cause the next action potential to come through. So let's look at how different axons take on the task of sending action potentials. Because depending on how large that axon is, or the degree of myelination is going to tell you how fast something is going to happen. So these large diameter fibers, they have less resistance. So they can send information very quickly. So the larger the fiber, the faster the information gets sent. The larger the fiber, the faster the information gets sent. Do we have myelination? Or not? That's also going to depend on how fast something gets sent along. We have continuous conduction. That's an non-myelinated axon. So that axon of that nerve is it's not myelinated, so that information is not going to jump anything. It's just going to continue to travel down that axon. It takes longer because it's depolarizing and repolarizing at every single part. Saltatory conduction is 30 times faster. That is because we are jumping from gap to gap, like I showed before. We have our axon. We have our myelin. We have our myelinated gaps. So that action potential jumps, 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 jumps. So it goes so much faster. Those action potentials only occur at the gaps. And it just jumps from one gap to the next. That's why it is so much quicker to get information from one cell or from across that axon to get it back to the nervous system much faster. So an action potential propagation, as we've talked about, it's sending that information along. So if we get information at this dendrite up here, it's going to send that information along. That is propagation. These non-myelinated axons, well, we get a stimulus here. It's going to send it over to the next spot where that ion channels continue to open. It's going to go down here where the next ion channel is going to open, down to here where the next ion channel is going to open, and so on, and so on, and so on. But these myelin sheaths, look, up, we jump to the next gap. We jump to the next gap. It just, that action potential happens just at the gap. I'm sure most of you have heard, at least heard of multiple sclerosis. It's an autoimmune disease. Autoimmune means your own immune cells attack your body. Well, what it's your immune system is attacking is that myelin on those nerve axons. So multiple sclerosis means that myelin is turning into sclerosis or hardens. So information either gets to the brain a lot slower or it just kind of ceases to happen. So information that starts off nice and strong just kind of dies off. And it causes demyelinated axons to have more sodium channels. And it causes relapse and remission, meaning symptoms go away for a while, then they come back again. Symptoms go away for a while, then they come back again. You get some kind of weird visual disturbances, weakness, not able to pick things up well. You can't control your muscles as well. You can't talk as well. Even get incontinence. They do have some pretty good drugs out there for this now. There's even shots. I have a friend with MS and he gets shots and it's completely put him into remission. So he can go about his daily life. But some research is showing that if we keep high levels of vitamin D in our system, it can reduce the risk of developing multiple sclerosis. So this is some newer research that's out. That's kind of cool. So another reason to take vitamin D supplements just besides living in the Northern Hemisphere. So we have different kinds of nerve fibers. We've kind of talked about it with large and small neurons. Well, let's look at it a little more. So we have group A fibers. These group A fibers, of course, are going to be the largest. And we talked about how large fibers are myelinated. They're somatic, sensory, and motor fibers. Skin, muscles, and joints. They can send information at 300 miles an hour. That's how fast something gets sent to your brain and sent back out. Group B fibers, they're not as big as group A fibers. They're considered intermediate diameter compared to A and C. They're lightly myelinated, so information still travels quick, but not as quick. Only about 30 miles an hour. Our group C fibers, they're the smallest. We kind of talked about those, the small unmyelinated fibers. Information goes at 2 miles per hour. If you've ever walked on a treadmill at 2 miles per hour, it's just gives you a good idea of how fast this information gets up to your brain. It's kind of interesting. It's just to kind of give you an idea. but they're usually autonomic nervous system, visceral motor, so going to like smooth muscle, sensory fibers from visceral organs, those are group B and group C fibers specifically. So what happens if we end up with impaired action potential propagation? That means information is not getting sent. We get an action potential started, but it doesn't travel down the axon correctly. It could be caused from chemical or physical problems, like anesthetics. Anesthetics will block those voltage-gated sodium channels from opening. That's what can cause something like cold temperatures. That can cause the blood vessels in your fingers or your toes to constrict. They get smaller. Blood doesn't travel through. And when that happens, we don't get blood to our oxygen, or blood and oxygen to our neurons. That's where you kind of get that goes to sleep feeling. That tingling sensation, goes numb, or you can't feel anything. That's all. That's a action potentials can't get sent along because it's not getting neurons are not getting the information that they need. So let's look at a synapse. I showed you before what is happening through a nerve, an neuron. So a synapse is where one neuron is communicating with the other. Where I had one neuron drawn out and the next neuron drawn out. Well, this is my presynaptic and my postsynaptic neuron. Because presynaptic, it sends information. My postsynaptic is what receives information. That is just the name for those two. So here's some other pictures that show what I just drew. We have what are called axosomatic synapses. We have an axon that attaches to the nerve body. Axodendritic. We have the axon that attaches to the dendrites of this neuron. Axonal synapse is where an axon attaches to an axon. And that's the axon hillock. So it's still technically considered the axon at this point. So here's a really cool electron micrograph of different neurons communicating with each other. So I just labeled all of those: axodendritic, axon terminal, and dendrites, axosomatic, axon terminal in the cell body. Those are the more common, but we have axonal, axon to axon, dendritic, dendrite to dendrite, somatodendritic, dendrite to soma, cell body. And then we have chemical synapses and electrical synapses. Chemical. That's where we use that neurotransmitter to communicate with one cell to the next. That is where we have our synaptic vesicles. Remember those synaptic vesicles? They're filled with neurotransmitters in the presynaptic neuron. And on that postsynaptic neuron, we have a receptor region for that. So that is where that postsynaptic neuron is going to receive that neurotransmitter to transfer the action potential. And then that synaptic cleft. That synaptic cleft is this area right here. That is the space between the two nerves, however it may be done. At this synapse between the two neurons, we have an electrical impulse that starts. The neurons communicate from one cell to the next through chemicals. Then it turns back to electrical to travel down that axon or down that dendrite, wherever it needs to go. So at that synaptic cleft, that synaptic cleft prevents nerve impulses from directly passing from one neuron to the next, even chemical. It makes sure transmission is one direction, unidirectional. It only goes one way. We need that communication happening. It needs to go one direction. So here's this chemical synapse. I drew it all out earlier on that when I opened up that blank slide. That action potential comes, it causes the calcium channels to open, calcium enters, calcium causes those synaptic vesicles to release neurotransmitter. That neurotransmitter goes across that synaptic cleft and binds to the postsynaptic membrane on gated ion channels. That neurotransmitter opens those channels, which allows that sodium to come flying in, which causes depolarization of the cell. Once that neurotransmitter has to be broken off of that ion channel for that ion channel to close. Because as long as that neurotransmitter is bound, it's going to stay open. It's going to stay there until something comes in and terminates it. Well, then what happens to that neurotransmitter? That neurotransmitter could be taken up by astrocytes. Remember those cells that help monitor the central nervous system? Or they can get taken back up into that axon terminal of the original neuron, that presynaptic neuron, to be used again. We have enzymes that degrade it, like acetylcholinesterase in our muscles. That acetylcholinesterase degrades acetylcholine, so it can't cause that acetylcholine to attach to another channel and open the channel when it doesn't need to be. They also diffuse away from that synaptic cleft. We don't need that neurotransmitter to stick in there and continue to cause problems by sending off something that was never sent in the first place, sending off another action potential that may not have ever needed to be sent off. So here's this picture with everything that is occurring. We have that action potential coming in, which causes those voltage gated calcium channels to open. Calcium channels cause the synaptic vesicles to release their neurotransmitter. The neurotransmitter binds to the ion channels. The ions flow in and out until that neurotransmitter gets released off, and it either gets broken up by enzymes, diffused away, or gets retaken up by the original neuron. So there is a little bit of a delay. Once that neurotransmitter's been released, it takes time for that neurotransmitter to diffuse across that synaptic cleft and to bind to that receptor. I mean, it's only 0.3 to 5 milliseconds. It's nothing really noticeable, but there is a little bit of a delay, and that is considered a synaptic delay. And that is the one rate limiting step, because nothing can happen until that neurotransmitter actually binds. Because the actual potential being sent is very quickly. It happens very, very quickly. But we have that synaptic delay because from one neuron to talk to the next neuron, it takes time to diffuse that neurotransmitter across. That's all a synaptic delay really is. So we do have some electrical synapses, but they're not common at all. They're very, because neurons communicate through electricity. So they're usually joined by some kind of gap junction because that's how they can communicate. And that happens between adjacent neurons. But this is where information can be one direction or multiple or bidirectional, so two directions. It happens in the brain. Because for eye movement, sometimes your eyes have to go different ways. Or our hippocampus. And that's where emotions and memories are stored. So something that reminds you of something that makes you sad, that's your hippocampus doing that. Electrical synapses are found mostly in embryonic nerve tissue. So as we're developing, this is what's out there. So our neurotransmitters, when they bind to that postsynaptic membrane, they cause graded potentials. But it's based on how much neurotransmitter is released and how long does that neurotransmitter stay in that synaptic cleft between the two neurons. That all depends. That depends on how graded that potential is. And we have two different kinds of potentials. We can have an excitatory postsynaptic potential or an inhibitory postsynaptic potential. So it can excite and cause something or it can inhibit something from happening. So here's an excitatory. So it's causing something. It says fire, do something. Versus the inhibitory says don't fire, don't do something. So these different neurons can cause different responses. So let's look at these excitatory synapses. So in an excitatory synapse, our neurotransmitter is going to bind and it's going to open those gates. Sodium and potassium are going to flow in opposite directions. Sodium's going to fly into the cell first. Potassium is going to fly out. But that sodium going in is a lot more than that potassium coming out. So that's an excitatory postsynaptic potential. More sodium is going in than potassium going out. And an inhibitory, we have potassium channels open and moving out of the cell. We have chlorine channels open and it moves out of the cell. Because our cell becomes more negative. It moves further away from threshold. We're getting rid of any positive, but we're also getting rid of more negative. So it causes a hyperpolarization. Inhibitory causes a hyperpolarization. Our cell becomes more negative. An excitatory, a single excitatory postsynaptic potential cannot induce an action potential, but they can summate, which means multiple coming in. They can influence that postsynaptic neuron. But an inhibitory postsynaptic potential, those can also summate. Means many of them coming in together. Because our neurons will receive excitatory and inhibitory input from neurons around them. So only if excitatory predominates, so we're getting more excitatory than inhibitory, that causes that action potential to be generated. Because we usually get excitatory and inhibitory for things. So we need more excitatory than inhibitory to cause it to happen. So we have temporal and spatial summations. So in a temporal summation, we usually have a rapid fire of one or more presynaptic neurons that are sending out. That first impulse is excitatory, but then another excitatory comes. So that adds to the first. So we have an excitatory, another excitatory, another excitatory. So a spatial summation. We have a lot of receptors that are activated. So we're getting excitatory from different places. So an excitatory is coming from here. An excitatory is coming from here. An excitatory is coming from here. So they're coming from a lot of different places to be added together. That is spatial. Versus temporal is coming from the same place. 1, 2, 3. Spatial is coming from different places. So this chart just shows what we just talked about. Let's look at synaptic potentiation. So if we keep using the same synapse, it'll cause that postsynaptic cell to excite more. Because if that same synapse, we keep getting calcium into that presynaptic terminal, which will release that neurotransmitter, the more that happens, it leads to more excitatory postsynaptic neurons. Because that potentiation causes those calcium voltage gates to open. Calcium also activates kinase enzymes because it leads to subsequent stimuli. So this is how our long-term memory and learning occurs, is synaptic potentiation. What about presynaptic inhibition? So if we release an excitatory neurotransmitter, but it gets inhibited by another neuron, by an axo-axonal synapse, that inhibits that presynaptic excitatory response because another neuron is saying, hey, no, we don't need that. It causes to less neurotransmitter being released. So it's not as big of an excitatory response. So here is showing the graded potentials and action potentials, how they have short distances versus long distances. Graded potentials are short distances. Action potentials are those longer distances. Amplitude and a graded potential, they vary. They decay with distance versus that action potential is an all or none. Graded potentials use chemicals or sensory stimulus. Action potentials use some kind of voltage. Positive feedback is absent in a graded potential, but we have them in, or a graded potential, a positive feedback is absent. Action potential, it's present. The functioning of different channels in a graded potential versus an action potential. So let's take some time to look at neurotransmitters, because different neurotransmitters are used, and that is how our nervous system communicates. And we have more than 50, at least 50 more have probably been identified by now, since your book was written like 3 or 4 years ago was the last update. So who knows how many they've come across by now. So we usually make at least 2 neurotransmitters, sometimes more, because sometimes we have to inhibit things, sometimes we have to excite things, sometimes we have to start some things versus stopping something. So we need different neurotransmitters to do different goals. And they're kind of released based on the kind of stimulations our neurons are getting. So, acetylcholine. Acetylcholine, as we know, happens with muscles. We talked about it during muscles. It was the first one to be identified, and it's the one we understand the best. And as I said, neuromuscular junction, so it happens at muscles. It's also used by our autonomic nervous system. Some central nervous system will have to at neuromuscular junctions. It's made by acetic acid and choline. Acetic acid comes from the neuron cell bodies. Choline comes in to our neurons and gets put together with acetic acid to make acetylcholine by the choline acetyltransferase enzyme. It's degraded by the acetylcholinesterase, as we talked about with muscles. We also have different catecholamines that work as neurotransmitters. We have dopamine, epinephrine, norepinephrine. These all come from an amino acid named tyrosine. Indolamines, like serotonin. That's made from tryptophan, another amino acid. Histamine comes from the amino acid histidine. Some of them work for emotional behaviors. Your biological clock. Imbalances sometimes are what are associated with mental illness, because then our neurons can't work correctly, and that's what can cause a lot of these mental illnesses. So as we kind of said up here, a lot of these, like serotonin, comes from tryptophan amino acid. Histamine comes from histidine, another amino acid. Amino acids sometimes can be used as neurotransmitters, like glutamate, aspartate, glycine, GABA, or gamma aminobutyric acid. Those are all different amino acids that work as neurotransmitters. We have neuropeptides, strings of amino acids like substance P. Substance P lets you know something painful is happening. But then endorphins or reduce that pain perception. So like substance P and endorphins work opposite each other. We have somatostatin and cholecystokinin, help with digestion. We even have purines, which are parts of nucleic acids that can work as neurotransmitters. Adenosine, it's an inhibitor to the brain. Caffeine blocks adenosine receptors. Purines can cause calcium to go into those astrocytes. So some of these have positive effects, some of these have some negative effects. Gasotransmitters. Gaso, they're a gas like nitric oxide, carbon monoxide, hydrogen sulfide gas. As I said, not all of these are good. So they're going to bind with protein receptors. Nitric oxide, learning and formation of new memories. But it can also cause brain damage and strokes. Hydrogen sulfide gas. It alters the function of ion channels. Endocannabinoids. THC. That active ingredient in marijuana. They work in the brain. But our brain also synthesizes it on demand. It's thought to be involved with learning and memory. Neuronal development. Controls your appetite. Suppresses nausea. That's a good thing. Sometimes, depending on what's causing that nausea. So neurotransmitters do a lot of different things for a lot of different parts of our body and our brain. So they're grouped by effect and action. So neurotransmitters, as we've kind of looked at, they can be excitatory where they depolarize, or they can inhibit and hyperpolarize. Hyperpolarize means it's much harder to get that action potential started. GABA and glycine are inhibitory. Glutamate's excitatory. Acetylcholine and norepinephrine, they will bind to different receptors that have opposite effects. Acetylcholine can be excitatory to muscle, but it can be inhibitory to cardiac muscle. So direct action. A direct action is how I showed how that neurotransmitter binds directly to and opens that ion channel. Acetylcholine, a lot of amino acids do that. That's what happens between neurons to neurons and neurons to muscles. They can have an indirect, where they work through second messengers, usually some kind of G protein. Similar effects to hormones, because hormones have a very long-lasting effect. They can be neuromodulators, because they do not directly cause that excitatory or that inhibitory, but they can affect the strength of that synaptic transmission. They can influence what happens to that neurotransmitter being made. Once it's been secreted, what happens to it? Does it get taken back into the cell? Does it get degraded by enzymes? Does it diffuse away from the terminal? They can change the sensitivity of that postsynaptic membrane. Could be releases of paracrine, which only has a local effect. So as I've mentioned, a lot of them are ligand gated channels is how these neurotransmitters talk to their postsynaptic cell. Because once they bind to that ligand, it's immediate. So those excitatory receptors allow small cations like sodium to come flying into that cell. Or if they're inhibitory, they're going to allow chloride into that cell and hyperpolarize. That chlorine is negative. We have that negative 70. We allow chlorine in. Whoop. Now we're down to like negative 100, which makes it harder to get an extra potential started. So here's neurotransmitters working as a ligand. They bind to this channel, which allows them to open. But what about G protein linked receptors? Because they can be an indirect, complex, slow, sometimes they're prolonged effects. Because we have some kind of transmembrane protein. Those transmembrane proteins bind to both, you can do the outside of the cell and the inside of the cell. And they cause usually some kind of metabolic changes. Muscarinic acetylcholine receptors are some. We can have biogenic amines, neuropeptides. So what happens in a G protein linked receptor? That neurotransmitter is going to bind to the G protein receptor. It's going to activate a G protein. Once that G protein is activated, we use second messengers like cyclic ANP, cyclic GMP, diacylglycerol, even calcium is a second messenger. That opens or closes ion channels, can activate enzymes, phosphorylate channels, and it activates genes and induces protein synthesis of different kinds. So here's this whole process. So we get that neurotransmitter. Here it is. It is binding to that G protein. That G protein then activates that GTP. GTP comes over to that adenylate cyclase, which then, that GTP actually binds to the adenylate cyclase, and it converts ATP to CAMP. That CAMP allows these channels to open. So it's just a longer process to get the channels open with these G protein linked receptors than it is through a direct ligand, where they directly connect to the channel and open the channel. Just like muscles function in groups, neurons function in groups. Because there are billions of neurons in our central nervous system. So they have to work together somehow. So we as a human being function smoothly as a whole. A neuronal pool, that is that group of neurons working together. So within a neuronal pool, we have a discharge zone where neurons closer to that incoming fiber, they're more likely to send off an impulse. Where in the facilitated zone, they're further away. So it takes more to excite them to threshold. unless they get stimulated by something else. Something else could start them to be stimulated. So this is showing how this happens. We have this presynaptic input, and we have all these branches going out to several different neurons, this whole neuronal pool here, sending out all the same information to each of the different neurons. We have the facilitated zone and the discharge zone. So as information gets sent in, it goes along one pathway. And that one neuron stimulates the next neuron, which stimulates the next one, which stimulates the next one, and so on. And they work as an all or none manner to produce a response. Like how I showed this neuron out here gives us information that stimulates the next neuron. It'll stimulate the next neuron, and so on. Up to the brain, and it'll do the same thing back out. Our reflexes. You know how if you've ever been to the doctor where they tap that tendon, your patellar tendon below your patella, your kneecap, and it causes that effect? You to kind of kick your leg unconsciously? A reflex. It is an automatic response to a stimuli. What is happening is, say you get tapped with one of those those hammers at your patella, it activates this neuron, which goes to, let me get rid of some of this. So what happens here? This goes in to the spinal cord. Which goes to the next neuron, which will come out and produce a response. It literally goes into the spinal cord and comes right back out. That is that reflex arc. You get that sensory neuron that hits that receptor, goes into the central nervous system integration center. With a reflex, it just goes to the spinal cord. Then it comes back out as a motor neuron, and that's where your foot kicks. Just like this. Comes in, sensory information comes in, hits that spinal cord, and it comes back out, and you get the response from being hit with that reflex hammer. So in parallel processing, information travels through several different pathways, because we need more information being sent sometimes. Like a smell that's associated with an experience. You ever walked into a store or something and smelled something and it reminded you of something? Could be a person, could be a place. That is happening through parallel processing, and it's usually higher level mental functioning is where that all takes place. So what kind of circuits do we have where neurons communicate? We have diverging, converging, reverberating, and parallel after discharge. So here is our diverging. We start with one, it sends information to two, those two neurons send it to two more, those two neurons send it to two more. So that can actually activate hundreds to thousands of neurons that go to muscles. We have on convergent, we talked about this once before, we have three different inputs going to the same neuron. Like different sensory stimuli that can elicit the same memory. Like this, like a smell, the temperature outside, like like a bonfire, the cool air, the smell of the bonfire, all of it, people around you, all just kind of triggers a specific memory. That's what happens in a converging circuit. Reverberating. Well, we're sending information to one neuron, that's sending information to another neuron, that's sending information to another neuron, but oh, now we're coming back up and sending it through again. That's how we breathe. Because it's like, hey, breathe in. Oh, breathe in again, breathe out, breathe in, breathe out, breathe in. That's how we sleep and wake up. Walking. Think if this wasn't happening, like right foot, left foot, right foot, left foot, right foot, left foot. How well would we be able to walk if we didn't have a reverberating circuit? And here's a parallel circuit. Like one neuron sends out information to that 1, 2, 3, 4 neurons that all kind of eventually come back together to one neuron again. That can help you with math. If you're good at math, you probably have some really good parallel circuits. So where does our nervous system come from? Well, it starts from what is called a neural tube that comes from the ectoderm. I know we've looked at a lot of these different layers. Neural tube and neural crest comes from that ectoderm. Your neural tube becomes your central nervous system, so your brain and your spinal cord. So that neural tube creates a lot of cells. They become amniotic and they migrate. Their axons start to connect with different places to become neurons. We have what is called a growth cone. That is at the tip of the axons, and that allows it to interact with the environment around it. We get neurotropins that attack or repel this growth cone. We have nerve growth factor to keep these neuroblast cells alive as we're trying to create our nervous system. Filopodia is where that growth cone processes. They're going towards a specific target. So once this axon finds its target, could be a muscle, could be an organ, it could just be sensory. They have to make a synapse. Could be with the muscle, could be with that organ, it could be with another nerve. This is where those astrocytes kind of come in and help support it. Make sure it forms that synapse. Needs to get that cholesterol because that's what constructs some of these synapses. 2/3 of our neurons die before we're even born. That's crazy. Because if these axons don't synapse with the right place, they undergo apoptosis. While other ones just go through apoptosis through development. As we start learning things, as we grow, it makes those synapses stronger, or it prunes away ones that are no longer needed. There are some genes that promote excessive synapses. But it also this gene promotes too many synapses being pruned away. And that is what is thought to be schizophrenia. They're amniotic after birth. But some neurons continue to divide, like olfactory, your nerve, hippocampus, memory. Those are going to continue to divide. So here's this neuronal growth cone under an electron microscope. It looks pretty cool. So that is it for the basic information about the nervous system. The next chapter is we're going to spend the entire time talking about the central nervous system. That central nervous system, again, is just that brain and that spinal cord. So we will spend the next chapter talking about that. Thank you very much. Bye.